Good morning, everyone, and welcome to the third day of our online program to the Introduction to Microbiology and Biotechnology, organized by the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. So, on the behalf of this department as well as from the university, I welcome you all to our third session. And today, again, we have a very exciting speakers and talks. Two speakers, eminent speakers, we do have with us today from our department. Professor Somsubhra Thakur Choudhury is going to deliver his lecture, and another speaker we have Dr. Mohit Majumder, and Professor Thakur Choudhury will be speaking on the central dogma of molecular biology, a basic insight. And Dr. Mohit Majumdar will be speaking on the introduction to bioinformatics. So as I'm seeing that these two topics are very interesting and very much emerging areas of research in today's biotechnology and microbiological world. So I think you all will enjoy a lot for today's session and you will have a very good lesson, take home lesson from this session. So Professor Calcutta, are you there?
I'm there, Rajat sir.
Yes. So, if everything is fine, if the live streaming, we have the YouTube live streaming also going on with us.
Yes.
So, if that is going on very nicely, so it is almost 9:57. 3 minutes we do have on our hands. So, in these 3 minutes, I'm just trying to, talking about my partner. Um, for previous days, I'm speaking about the same thing, but still, because maybe some participants are coming newly in this session, so for them, I'm just telling that our department is a very vibrant department and very, I mean, uh, active department in the beautiful campus of Sister Nivedita University, Kolkata. I, Dr. Rajat Pal, is requesting you all to please visit at least once whenever you are visiting Kolkata, the beautiful city, the city of joy, and with the Sandesh and Mishti Doi, you can have a taste of our sister Nivedita University. Okay. So the Sister Nivedita University is a very, a new university, although, but still, we all faculty members and staff members and high officials, we all are trying, and with my lovely students, we all are trying hard to put this university forward and to go ahead. And I request you all that please do visit once at our university, at our department. We five faculties are there with me, Rajat Pal. There are four different other faculties. Professor Thakur Choudhury, who is going to speak today. Professor Calcutta, who has spoken in the first day. Professor Atre Ghosh, she will be speaking on the next day, tomorrow. And Professor Milan Boni, he will be speaking on the last day of this webinar. So we five are the faculty members of my department, and we are blessed to have Professor Dhrubajyoti Chattopadhyay as our Vice Chancellor. He is also from the same background, from microbiology and biotechnology background. So under his guidance, we are trying to run our department very smoothly. In our department, we generally offer both postgraduate and undergraduate courses on microbiology and biotechnology, as well as we have started our research for, offering the PhD degrees also. So this is nutshell of my department. Without wasting any more time, I will, invite Dr. Somsubhra Thakur Choudhury for today's lecture. He has done his graduation and post-graduation in microbiology, and he has done his PhD from Indian Institute of Chemical Biology, CSIR, Kolkata. And he, after that, he has joined as an assistant professor of the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. So I'm welcoming you, sir, in this webinar. Please go ahead and take the screen.
So, yeah, thank you. Thank you, Rajat sir. Am I audible clearly?
Yes, 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 sir, you
Okay,
are audible clearly.
Okay. Another thing I want to confirm that are my slides visible?
Yes, it is perfectly
Yes.
visible, sir.
One another thing. Is it there that my photo is coming aside the slide or not? Only the slides are coming.
No, only the slides are coming.
Okay. Thank you. Thank you, Roger sir. So I can start. I am at
Yeah.
just now. It is past 10. So good morning, students. Good morning, participants. Today I am going to talk on central dogma of molecular biology, a basic insight. Practically, I go through this session in our class. It takes almost 6 months, and I have to give the overview in 1 hour. So today is a test of mine, not yours. So without wasting much time, I want to go to the slides. And first, I want to mention that my topic is just linked with the previous talk, yesterday's talk that was given by Professor Roger Pal, and it is just a continuation of that. It is just a more exploration of that. So I will skip some slide at it has been already discussed yesterday. So if you have any doubt, you can send your question in the chat box. Okay, so let's start. As Roger said, described yesterday also that we have two types of nucleic acid in our body. One is DNA and RNA. There are four types of nitrogenous bases: thymine, cytosine, adenine, guanine present in DNA, and in RNA it is uracil, cytosine, adenine, and guanine. So whenever we are terming nucleic acid or deoxyribonucleic acid, the first thing that comes is the nitrogenous bases. There are two types of nitrogenous bases: purines and pyrimidines. A simple thing to understand, a simple thing to remember, that when the name is small, the structure is bigger. So purine is a smaller name, and there are two types of purine, that is adenine and guanine, and their structures are bigger. There are three pyrimidine molecules. Pyrimidine is a bigger name, the structure will be smaller. There are cytosine, thymine, and uracil. So after the nitrogenous base, if you can remember the full form of deoxyribonucleic acid, after the nucleic acid, before the nucleic acid, comes the term that is ribo. Ribo term comes from the sugar molecule, that is ribose, which is a pentose sugar molecule. So with the nitrogenous base, the sugar remain attached, and when the nitrogenous base and sugar remain attached, we call it nucleoside. And there is another component, that is phosphate. Whenever with the nitrogenous base, sugar and phosphate is present, we call it nucleotide. You can see here the base, the sugar, and the phosphate is present. But in our body, normally the nucleotides doesn't remain with a single sugar. It remains with the three sugar. Three means tri. So we call it nucleoside triphosphate. Roger has already mentioned yesterday that when in the ribose sugar, we term the carbon numbers as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. When the 2 prime carbon, the OH is present, we call it a ribonucleoside triphosphate, or RNTP, that makes the RNA. And whenever H is present, the O gets eliminated. That is oxygen has get eliminated, so we call it deoxyribonucleoside triphosphate, or DNTPs, which joins together to form a DNA chain. So DNA chain is just like a long string where beads of nucleotides are attached with each other. So how the beads of nucleotides get attached with each other? So I will come to that, and that is very interesting. So we have to attach these nucleotides with one another. So first we need two nucleotides. So we have two nucleotides. And here you can see in the 3 prime position there is OH, and oxygen has two lone pairs of electron. And whenever there is a molecule which have lone pair of electron, it is very much prone for the reaction. So free electron of oxygen attacks the first phosphate. I have said there are three phosphate. First phosphate is called alpha, second phosphate is called beta, and the third phosphate is called gamma. So oxygen attacks the alpha phosphate, and the bond between oxygen and phosphate breaks. So the oxygen of the previous nucleoside triphosphate now attached with the phosphate of the second nucleotide triphosphate. So what happens? So by this bond, this OPO bond, the two nucleotides has attached with one another, and the PPI, that means the 2-phosphate, we call it pyrophosphate, gets eliminated. So whenever a bond O bond is present, we call is ester bond, bond O bond. So there are two ester bonds, so we can call it diester bond. And between these two ester bond, there is a phosphate group is present, so we can call it phosphodiester bond. So now this is how the two nucleotides get attached with one another. Now in a DNA, there are very numerous numbers of nucleotides which are attached with one another. So for example, I am taking 4 nucleotides. So what will happen? It will form this chain. And here you can see that where what the nucleotides are attached with each other by a phosphodiester bond. And here you can see that only at the first nucleotide, at the 5' position, triphosphate is present. And at the last nucleotide, at the 3' position, OH is present. So only there will be one nucleotide where the triphosphate will remain present. So we call it at the 5' end. So we call it a 5' end of the DNA chain. And the 3' end, there will be a OH will remain present. So we will call it a 3' end. Now a very important thing, that is, I want that everybody remember this thing, that to add a new nucleotide triphosphate or nucleotide, we need a pre-existing OH, 3' OH. So if the 3' OH is not present, then the oxygen cannot attack the alpha phosphate and one nucleotide cannot join with the next one. So to join one nucleotide with another nucleotide, we need a pre-existing 3' OH. Another thing is that the DNA or RNA synthesis the direction of DNA and RNA synthesis is always from 5' end to 3' end. You can see that the DNA synthesis and RNA synthesis have taken from 5' end to the 3' end. And if the new nucleotide will join, it will join to the last previously present last nucleotide, and the chain will grow from the 5' direction to the 3' direction only. So this is the DNA structure that Rajasthan has already mentioned. I just want to give an interesting calculation that I think that will make you amazed, make you surprised. That in our human cell, in our single human cell, there are 3 into 10 to the power 9 number of base pairs in a haploid set of chromosomes. What is haploid set? We are diploid animals. We have in each of our cell, we have 2 sets of chromosome. If we consider one set, we will call it haploid set. In a haploid set, in a single cell, in a haploid set, there are 3 into 10 to the power 9 base pairs present. And you can see the distance between two base pair is 3.32 angstrom or 3.4 angstrom. I am considering 3.4 angstrom. So distance between two base pairs is 3.4 angstrom. So what is the total length of a haploid DNA? So total length of a haploid DNA is 3.4 into 3 into 10 to the power 9 or 10 into 10 to the power 9 or 1 meter. 10 to the power 10 angstrom means 1 meter. So length of a haploid set of DNA is 1 meter. So each our cell are diploid. So if we take the two DNA and we join together, it will cover total length of 2 meter. The DNA of one cell covers the length of 2 meter. And how many cells we have? In an average human body, we have 30 trillion cells. That is 30 into 10 to the power 12. So if we collect DNA of all the cells and join with one another, then what should be the distance? 2 for each cell, 30 into 10 to the power 12 cells. So 30 into 10 to the power 12 into 2 equal to 6 into 10 to the power 13, or 6, that is meter, or 6 into 10 to the power 10 kilometer. Can you imagine how much long it is? Say for an example, the distance between Earth and Sun is 150 million kilometer. That is 1.5 into 10 to the power 8 kilometer. So we divide it. If we divide it, 6 into 10 to the power 10 divided by 1.5 into 10 to the power 8, that comes 400. That is, if we collect DNA of all the cells of our average human body and if we join together, we can go to the Sun 400 times. Isn't it amazing? So we are now dealing, you are aspiring bio students. So you are dealing with a subject which can give you this wonder. Remember that first. So now let's come to our central dogma. My focus of today's lecture, that is central dogma. What is central dogma? Central dogma is the DNA is present in our cell, and from DNA the protein production takes place. So the process through which from DNA protein production takes place is known as central dogma. So there are 3 steps. First is replication, where from DNA another DNA synthesis takes place through an enzyme known as DNA polymerase. So DNA replication. What do you mean by replica? Replica means a same copy of another thing. If you have a one thing, if you want to make the replica of the same thing, you have to make the same copy, same pi to pi detailed thing of the previous thing. So it is called replica, and DNA replicates itself. DNA forms the same exact copy of its DNA, and this process is known as DNA replication. So this is, I think, this is a bit difficult thing. I think I will try to understand. I will try to go through it in quick time. Hopefully, you will understand. So previously, there was 3 hypotheses that how from one DNA another DNA gets produced. As we all know, DNA is double-stranded. So there may be 3 options. That is, first is semi-conservative. That is, if there is 2 parental strands, there is 2 DNA strand that is present in a parent body, or there is that is present previously. First of all, DNA replication takes place during cell division. So if there are 2 DNA strands present, whenever the replica will produce, one thing that can happen, that from one previous strand a new DNA strand will synthesize, and from another previous strand another new DNA strand will synthesize. Here in the picture, you can see previous DNA strands in blue and newly synthesized strands are in red. So one thing that may happen occur, that from one blue, one red has been prepared, and from one blue, another red has been prepared. This is called semi-conservative. Semi means half, because you can see in the new DNA, one part, half part is from previous one. So semi-conservative, half of the DNA has been conserved and half new DNA has been synthesized. There can be conservative. That means the previous DNA remains intact and the same copy of the DNA has been synthesized newly. So the previous DNA is intact and the complete new copy of DNA has been synthesized. That is conservative. There may be dispersive. That means some part of previous DNA is there, some part of newly synthesized DNA is there. So there were 3 hypotheses. 2 scientists, famous scientists, one is known as Messelson and another is known as Stahl, proved that DNA replication takes place in semi-conservative model. This is a long experiment. To cut short, what they did, they took E. coli, they cultured E. coli in a heavy nitrogen containing medium. That means where the nitrogen is N15. That means they cultured the E. coli in a N15 medium. So now all the DNA have heavy nitrogen in it. That means all the DNA are heavy. We have seen the nitrogen is present in the nitrogenous base. So all the nitrogenous bases that contains nitrogen will be heavy nitrogen. So the DNA will be heavy. We will mention it as H, and thereafter they took the cell and they continued the culture for one generation. That means for 20 minutes, where in 20 minutes one cellular division takes place in E. coli. So for 20 minutes they cultured in light DNA containing medium. So the newly synthesized DNA will contain the light nitrogen, or L. We will mention it L. That means where the nitrogen is 14. N14 is present, we will mention it as N. So here you can see that, as I have mentioned, the previous strand, the uh, original strand, as it was cultured in heavy nitrogen containing medium, both strands are HH, and whenever they culture one generation for low nitrogen containing medium, what happens is that the newly synthesized strand are L, that means light. So if it is conservative mode of replication, and then they collected the DNA and done a cesium chloride centrifugation, where the higher, that is the heavy DNA, will come in the um, lower part of the test tube, and the light DNA will come at the upper part of the test tube. Here you can see that if it is a semi-conservative mode of replication, then what should occur? A band should come at the lower part for the heavy DNA, and a band should come at the upper part for the light DNA. In the semi-conservative mode of replication, it should occur in the conservative mode of replication, whereas in the semi-conservative mode of replication, both of the DNA is a hybrid of heavy and light. So band should neither come to the heavy part or the upper part of the cesium chloride, nor the lower part of the cesium chloride. It should come in the middle for the heavy and light hybrid. And for the dispersive mode of replication, it is also same. It should come in the middle. So when they done the cesium chloride centrifugation, they found that the first thing, that is, two bands are not coming. One band is coming, and that is coming at the mid portion of the cesium chloride. So they became sure that the DNA replication doesn't take place in conservative manner. It should take place in semi-conservative or dispersive manner. Then which one is correct? Which one really occurs, semi-conservative or dispersive? For to confirm that, they run another generation in light medium, that is light nitrogen containing medium. They continued the growth for another generation, another 20 minutes in N14 containing medium. So what will occur? In N14 containing medium, you can understand that if you see the semi-conservative mode of replication, you will see that two DNA will be there which will be hybrid, and two DNA will be there which will be completely low molecule, that is lower molecular weight, that is lower density, LL. So there, whenever we done the cesium chloride centrifugation, we should get two band. One is at the medium, another is at the top. The medium is for heavy and low hybrid, which is, there are two DNA which are heavy and low hybrid, and we will get another band at low, that will get at the upper part of the cesium chloride. But in the dispersive mode of replication, there should come one band which is at the medium, because here the mixing are taking place between the parental DNA and the newly synthesizing DNA. And they found that properly, that what the middle, in the middle portion we are showing, they found two bands, and one is at the upper part of the cesium chloride, another is at the middle part of the cesium chloride, and it confirms that the DNA replication takes place in the semi-conservative model. That means from one parental strand, one newly synthesized DNA, one new DNA strand gets synthesized, and from another parental strand, another new DNA strand gets synthesized. So how the replication takes place? There are mainly 6 enzymes: helicase, SSB protein, topoisomerase, DNA polymerase, DNA primase, and DNA ligase. What are the functions of each? I will come one by one. First of helicase. What is helicase? DNA, whenever there is a term ase, that means it breaks. Helicase means it breaks the helix. So DNA remains in a helical form, as I have shown. So what will be the function of helicase? It is a ATP dependent enzyme and it will break the helix. Okay. Then comes another part, that is single stranded binding protein. Whenever DNA becomes free, whenever DNA becomes single stranded, there is a chance that it can rewind again and form double stranded. So we have to protect the reassociation of the two strands, and it is done by a protein known as single stranded DNA binding protein. From the name you can understand it binds with the single stranded DNA and it protects the re-annealing of the DNA. Okay. Then comes another important protein, that is topoisomerase. Hello, am I visible? Am I visible to everybody?
STC sir, you are visible, but your slides are not
Okay,
visible.
I just want that. So students, can you see that? One second. So can you see that? Say I have prepared this. This is a DNA. You can see that two DNA strands are there. One I have colored violet and one is white. And this is just like a DNA which are in a double helix. So what will occur whenever the replication will take place? So for an example, what will occur? Helicase will unwind this, and whenever helicase will unwind this, you can see a knot has been formed here. So if you don't open up the knot, you cannot further unwind the DNA. So a knot has been formed here, you can see that. So there should be an enzyme that opens this knot, and this is done by an enzyme known as topoisomerase. So let's go to the slide again. So as I was saying, whenever helicase was unwinding the DNA, a knot forms, and this knot is cut and joined again to release from this knot, and this is done by an enzyme known as topoisomerase. Okay, so there are the three proteins which have helped in the unwinding of the DNA, but up to now, new DNA synthesis hasn't taken place. Okay, so if we see this what has I have described now, the single stranded DNA, that is known as parental strand or template strand. Why we call it template? Because from this strand the new DNA strand will get synthesized, so it will act as a template, so we call it parental strand or the template strand. Now if we focus on a part of this parental strand, we can see that this is a single stranded DNA, so it should look like this. Okay, and what should occur? The corresponding bases, that is T should come and bind with A, G should come and bind with C, and it should form like this, and a new DNA strand should get synthesized, where the new DNTP should bind as I have mentioned earlier, by forming a phosphodiester bond. Is it okay? So this newly synthesized DNA strand, we will call it a daughter strand. This is the structure of DNA polymerase. It looks like a hand-like structure, where you can see new DNTPs, incoming nucleotides are coming, and by the phosphodiester bond formation, it gets attached with the newly synthesizing DNA strand. But first, it comes the first problem that I mentioned in the almost the first or second slide, that DNA polymerase cannot add nucleotide when there is, or cannot add nucleotide when there is no pre-existing 3' OH. It can only add nucleotide when there is existing 3' OH. But you can understand that for the first nucleotide it is adding, there is no 3' OH prior to that. Previously there is no 3' OH. So how it can add the first nucleotide? DNA polymerase cannot add the first nucleotide. And to solve this problem, we need an enzyme known as DNA primase or only primase. What occurs? Primase forms a part complementary of the DNA, but it is not a DNA, it is a RNA. So DNA primase forms an RNA section, and after forming a RNA section which is about 10 to 20 nucleotide long, it gets released. And whenever it gets, we call it RNA primer. Whenever it gets released, then you can see then the 3' OH is free, and now the DNA polymerase can come and starts its work. So now the DNA polymerase come and synthesizes the rest part of the DNA. So now we are getting a DNA where the first few nucleotides are RNA, and thereafter the total nucleotides are DNA. So another important thing, that DNA polymers also have proofreading activity. I always mention to my students that DNA is like the Bible or the main book where everything is written about myself. So a cell doesn't want, whenever it is replicating, doesn't want any mistake in the DNA. DNA replication is almost no very few mistakes takes place during DNA replication. And who plays a major role? That is the DNA polymerase, because DNA polymerase have a very impressive activity that is known as proofreading activity. What is proofreading? When we are when we are writing something, we goes and check it that we have written it right or wrong. The DNA polymerase do the same thing. Whenever it adds a wrong nucleotide, it understands that I have added a wrong nucleotide, I have to remove it. Then the DNA, the newly synthesized DNA strand, goes to another site of the enzyme which has exonuclease activity, which have 3' to 5' exonuclease activity, and it cleaves the nucleotide. And thereafter, it again goes to the polymerization site where the correct nucleotide gets added, so that the chance of addition of a wrong nucleotide becomes very low. Now, here you can see it is called a replication fork. Here you can see that, say it is the strand from, you can see the upper strand where a new DNA synthesis is taking place. The green one is RNA primer that I have mentioned, and this red one is the newly synthesized DNA. So here you can see the newly synthesized DNA is running from 5' to 3' direction. I have said in the second slide that DNA synthesis should always run from 5' to 3' direction. So here it runs from 5' to 3' direction, and we call it a leading strand. But what about the another strand? For the another strand, for the 5' to 3' synthesis, 5' to 3' synthesis, it should run from this direction to this direction. That means from right to left. And what occurs is that how we can synthesize the lagging strand continuously. We cannot synthesize the lagging strand continuously. Why? Because we cannot synthesize the lagging strand continuously. Say it is the DNA. So first the helicase unwinds some part of the DNA. So if it is the leading strand, here the DNA synthesis can take place continuously. But here I have to synthesize from this direction. So here primer binds or primase binds. It forms a short nucleotide of primer, and DNA polymerase synthesizes the rest part. Now the helicase had unwinded another part of the DNA. Now, whenever the direction is taking place from this side to this side, it can synthesize the newly strand continuously. But for this case, this part of DNA has been synthesized. Again, the rest part, again the primer has to bind here. It has to synthesize a small primase will bind here. It will synthesize a small primer, and again DNA polymerase will synthesize the rest part. So you can see that in this leading strand, the DNA synthesis will continue. It will newly synthesized DNA will be continuous, whereas in this case it will be fragmented. This is one fragment. This is another fragment. So in the lagging strand, in another strand, the DNA synthesis will be in fragments. So here you can see that in one strand it will be continuous, in another strand it will be fragmented, and these fragments are known as Okazaki fragments. Okay. So in the you can see that in the lagging strand, the DNA the DNA gets synthesized in fragments, where the first part is primer, then it is DNA. Thereafter a primer, then it is DNA. And primers are what? Primers are RNA. We cannot allow RNA in the DNA, so we have to remove from these primers. So which enzyme takes place? An enzyme that is known as RNase. I have said that when there is ase, it means it will cut. So RNase means it will cut RNA. So RNase cuts the RNA, and here you can see that if RNase cuts the RNA, for this is the DNA and this is the 3' part which have free OH. So from this free OH, DNA polymerase can again synthesize the rest part as DNA, but it cannot join the last part, like cannot join the last two nucleotides, because in the first nucleotide that is written just before, that is present just before the red DNA, red arrow, it contains only a single phosphate, because the beta and gamma phosphate has already been removed whenever it was synthesizing the primer. So I have already said that DNA polymerase is a enzyme which cannot join if there is no, if that the 3-phosphate is not present at 5' end. So it cannot join this nick, and this nick is sealed by an enzyme known as DNA ligase, which utilizes ATP and seals this nick. So we are done with the replication. And where the replication takes place? Replication takes place in the nucleus. Why in the nucleus? Because DNA is present in the nucleus. So you have to make a replica of the DNA. So you have to make it in the nucleus only. Then it comes the second step, where from the DNA RNA will form, and this process is known as transcription. And the major protein that plays an important role is RNA polymerase. So what happens? In the simple thing, you can see this is a DNA duplex, and this is the ash in color, and from DNA duplex the green colored RNA has formed. I have always said that the replication or transcription goes to 5' to 3' end. Here also you can see that RNA synthesis is taking place from 5' to 3' direction. So it will utilize from RNA synthesis, for the transcription, it will not utilize the both strand of DNA. It will utilize the only one strand of DNA that runs from 3' to 5' end. So it will utilize the DNA that runs from 3' to 5' end, and from that it will produce the RNA which is from 5' to 3' direction. So it will use DNA strand which runs from 3' to 5' direction. We will call it a template strand, and it will synthesize RNA which synthesizes from 5' to 3' direction. Okay, so this is also same thing that I have described earlier. Only the cases here in the DNA that the deoxyribonucleotide comes, and here the ribonucleotide triphosphate comes, and the same thing occurs, that is the OH attacks the alpha phosphate and the pyrophosphate releases and the new nucleotide joins with the previous one. Okay, so I am not going in details. So RNA polymerase plays an important role in transcription. In transcription, there are three stages: initiation, elongation, and termination. So initiation means from a one position, from a specific position of the RNA, from the DNA, from a specific position of the DNA, the RNA synthesis will initiate. So how it will understand that from which portion of the DNA I have to start the RNA synthesis? So to mention that, to point out that this is the portion from where you have to start the RNA synthesis, there is a part known as promoter. So in the DNA, there is a part known as promoter. And why it is called promoter? Because there are two specific regions in the DNA. That one is called -10 region, another is called -35 region. And these two regions makes the promoter. At the -10 region, there is a Tata at sequence, and -35 region, there is TT GACA sequence. TT GACA means it is not a name. There is thymine, thymine, guanine, adenine, cytosine, adenine. So it is TT GACA, and Tata thymine, adenine, thymine, adenine, adenine, thymine. So it is Tata at. So these, whenever RNA polymerase finds these two sequences, it understands that from the 10 nucleotide next, I have to start my RNA. I have to start my RNA synthesis process. So RNA polymerase is like a crab claw structure. And RNA polymerase, the deficit that RNA polymerase cannot identify the promoter sequence. To identify the promoter sequence, RNA polymerase needs another protein that is known as sigma factor. So whenever with core RNA polymerase, the sigma factor binds, it becomes a hollow enzyme. That means it becomes an active enzyme. Now it can continue the transcription process. And what happens? So here at the top you can see with the core RNA polymerase, the sigma factor has associated and it has formed a hollow enzyme. Whenever it has formed a hollow enzyme, then double-stranded DNA comes, from where the RNA synthesis will take place. And double-stranded DNA binds with the RNA polymerase, and the promoter sequence gets recognized by the sigma factor. Okay, now the sigma factor have recognized the promoter sequence. The double-stranded DNA has become associated with the RNA polymerase. But to form the RNA, we have to unwind the DNA. We have to unwind the DNA so that from one strand RNA synthesis takes place. But you can see here the DNA is not opened up. Here the DNA is closed. So this complex we will call a closed complex. Thereafter you can see that here one part of the DNA has already opened up. You can see a part of the DNA has become opened up. In the open complex formation, you can see a part of the DNA has opened up. So when that is a part of the DNA has opened up, and we call it a open complex. So first it was a closed complex. When DNA was in a double helical form only, it was not opened up, and after that it has opened up. We call it open complex, and we call this opened up junction as a transcription bubble. Then what happens? Then for RNA polymerase synthesizes a part of the RNA, but until RNA polymerase can synthesize more than 10 RNTPs together, it cannot continue the transcription procedure. So for various times, it starts the transcription and aborts. It starts the transcription and aborts. And whenever it can synthesize more than 10 RNTPs, it can add more than 10 RNTPs one after another correctly or one after another firmly, then it continues to the elongation process. And whenever it had added, you can see here it has added 4 more than 10 nucleotides together, and we will call it a initial transcription complex. We will call it a initial transcription complex. That means transcription initiation has taken place. Whenever transcription initiation has taken place, there is no need of the sigma factor, because sigma factor is only required for the recognition of the promoter. The promoter recognition has taken place, the transcription initiation has taken place. So the sigma factor gets removed. And now the RNA polymerase can itself continue or elongate the uh, RNA synthesis. So transcription elongation complex formation, and it starts synthesizing the RNA. And what happens whenever it comes to the termination sequence, whenever RNA polymerase comes to the termination sequence, the termination takes place and RNA gets separated from the DNA as well as RNA polymerase. So we have understand how the initiation takes place. Elongation means is just add one after another RNTPs. So there is nothing special in the elongation process, but there is special in the termination process, because termination takes place in two manners. One is row independent or factor independent termination. One is factor dependent or row dependent termination. Factor independent termination. Factor independent termination. From the name, you can understand that no factor is required. It is also known as intrinsic termination. And what occurs? How the intrinsic termination takes place? Whenever from the DNA the RNA gets synthesized. This the blue one is the RNA, the black one is the DNA. If you see the sequence in the blue one, then you will see that in the RNA there is a inverted repeat sequence. That means a sequence that there are two sequences that are complementary to each other in the RNA. So, and these inverted repeats are GC rich. So there are two sequences that are GC rich and they are inverted. So they can form a hairpin loop structure. That means they can, this in the RNA, normally we say that RNA is single stranded, but if there is a inverted repeat, RNA can also form a double stranded structure. And if RNA form a double stranded structure, after the inverted repeat, there is a sequence of long sequence of 20 nucleotides almost. There is a sequence, mane a sequence. So what happens whenever RNA forms the hairpin loop structure? You all know that between A and T there is double bond. Double Between A and T there is double bond, and double bond means it is a weak interaction. As hydrogen is after a weak interaction, if GC is present, then there are three triple hydrogen bonds which is much stronger, but between A and T there are two hydrogen bonds which is weaker. So whenever the RNA forms a hairpin loop structure, it gives a pull to the RNA that is associated with the DNA. So whenever RNA forms a hairpin loop structure, it gives a pull to the RNA that is associated with the DNA with A and T base pairing. A and T base pairing is loose, so it comes out from the DNA, and the RNA synthesis, RNA transcription completes, transcription terminates, RNA gets loosened out from the DNA. So this is row independent. It occurs through the intrinsic repeats. And what happens in a row dependent case? In row dependence case, in RNA, there is a sequence known as NUT site or row utilization site. So it is row dependent. So a protein that is row, that will come, and this whenever this RUT site gets synthesized, whenever this part of DNA, this part of RNA gets synthesized, where the RUT sequence is there, then a protein comes and binds with the RUT sequence that is known as row protein, and it moves towards the DNA RNA junction. That means where the RNA polymerase is there. So it moves towards the RNA polymerase, and this row protein has an important DNA RNA helicase activity. That means whenever DNA RNA is present, it can break the bonds between DNA and RNA. So what happens whenever it reaches the RNA polymerase by its DNA RNA helicase activity, it loses the RNA from the DNA, and now the RNA becomes free. So the termination has completed. It may be by the row independent manner, it may be by the row dependent manner. So now we have RNA, but we need some processing of the RNA, that is known as RNA processing or post-transcriptional modification. That means the modification that takes place after transcription. Why we need RNA modification? Because DNA was double stranded, which was very much safe, which is not very much uh, prone to damage. But RNA is single stranded, so RNA is much prone to damage. So we have to protect the RNA. So to protect the RNA, first thing that occurs, that means we have first thing that the two sides, that is the 5' end and the 3' end, we protect these two sides first. It is all our defense system. In war also, we first stops, or we first blocks the boundaries. So 5' and 3' are the boundaries. So we have to protect the boundaries first. There are two boundaries, 5' and 3' end. How we can protect the 5' boundary? In the 5' boundary, we add a 7-methyl guanosine, a specific nucleotide, that is 7-methyl guanosine, by a 5' linkage. It is very difficult to understand how the 5' linkage takes place, and it is also more difficult to make you understand in a short time. But you can just remember that at the 5' end of the RNA gets protected by a capping known as 7-methyl guanosine capping that protects the 5' end. And what is the function of the 7-methyl guanosine? First is protect the nascent DNA. Second, it also plays a important role during translation process. So 5' end is now protected. We have to protect the 3' end. So the 3' end, in the 3' end, a protein called polymerase adds a numerous number of adenine at the 3' end. How many adenines? 200 adenines. Poly A polymerase can add 200 adenine molecules at the 3' end of the RNA. So now we have a long RNA poly A tail. So if any damage takes place, that will not hamper the original RNA. Second, very important thing, that by this poly A tail, now what the transcription has taken place? Transcription has also taken place in the nucleus, because we have to do the transcription from the DNA, and DNA is present in nucleus. So we have to do the transcription in the nucleus also. But the next stage, that is translation, will take place in cytoplasm. So we have to move the RNA from the nucleus to cytoplasm. And for this, the poly A tail plays an important role. By this poly A tail, RNA moves from nucleus to the cytoplasm. It also plays an important role in translation. Next, another modification, that is RNA splicing. What is RNA splicing? Whenever RNA gets synthesized, from RNA the protein will be synthesized by translation procedure. But all the part of the RNA doesn't code for the protein. That means from the full part of RNA, the protein doesn't get synthesized. There are some part of RNA that is known as intron. Here you can see in orange part. That is known as intron, from which the protein synthesis doesn't take place. So we have to remove these introns. And this green part, from where the protein synthesis takes place, which codes for the protein. What is coding? I will come later. Which codes for the protein is known as exon. So intron doesn't code for the protein, exon codes for the protein. So the part that doesn't code for the protein, we have to remove that part. And we have to join the parts, that is exon, that codes for the protein together. And this is called RNA splicing. There's a very difficult procedure, that is spliceosome mechanism, through which the RNA splicing takes place. So now we have the mRNA, where in the 5' we have capping, we have the protein coding sequence, and at the 3' we have poly A tail. Now it is the complete mRNA, and it comes from nucleus to the cytoplasm. And in the cytoplasm, the translation takes place. Now, why it is translation? DNA means deoxyribonucleic acid. RNA means ribonucleic acid. So what is the language? You can understand the language is nucleic acid. The basic thing is nucleic acid. But in the translation procedure, we have to make protein or polypeptide. And the proteins are made of amino acid, as Rajat has said yesterday. So previously it was nucleic acid. Now from the nucleic acid we are making a thing that is amino acid. So if in the first two cases the language was nucleic acid and the language has changed. In this through this procedure we will change the language to amino acid. So whenever we change a language we say it translation. English to Bengali or English to anyone a language, we call it translation. So from the nucleic acid language, we are changing it to the amino acid language. So this procedure is known as translation. So first thing comes first. Then the nucleic acid, we all have understand by this time what is nucleic acid. Then how from nucleic acid amino acid will produce. So if it is a RNA sequence, the three nucleic acids that remains together, we call it a codon. The three nucleic acids in an RNA sequence that remains one after another, we call it a codon. And each codon codes for one amino acid. So there are 20 amino acids and there are 4 different types of nucleic acids. So if we make a combination and permutation of combination of 4 nucleic acids in 3 arrangement, we will get 64 arrangements. And each arrangement codes for each or more than one arrangement codes for one amino acid. Say, for example, if in the RNA we have AUG, it will code for methionine. That means methionine will come and bind here. If the code is GUC, then valine will come and bind here. If the code is AGU, then the serine will come here. And this is the codon table that where it has been shown that if there are 3 nucleotides in which combination codes for which amino acids. So and there are 3 codons are there which doesn't code for any amino acid. We call it a stop codon. They are given in red. They are UAA, UGA, and UAG. These codons doesn't code for any amino acid. They are known as stop codon. So what happens in a messenger RNA? In the messenger RNA, there are 3 important things. One is open reading frame. That is the part of mRNA from where the protein synthesis will take place. There is a start codon. That means here also we have to know from where we have to start the translation. So there is a start codon. The start codon is normally AUG codon, 5' to 3' AUG codon. GUG as well as UUG can also act as a start codon. And as I have mentioned earlier, there are 3 stop codon: UAG, UGA, and UAA. This was also has mentioned by Roger yesterday that what is the structure of tRNA. I am not going in details, but you can see here at the lower end of the tRNA there is known as anti-codon loop. So we have heard about codon. What is codon? A 3 nucleotide sequence that codes for a amino acid. Anti-codon means that is anti to the codon. That means that has complementary sequence to the codon. So by this anticodon loop, the tRNA is known as transfer RNA. Why it is called transfer RNA? I will come. So by this anticodon loop, the tRNA binds with the codon. And in the upper part, you can see there is acceptor stem or amino acid attachment site. That means if the codon is AUG, in the previous slide I have said that AUG codes for methionine, then the tRNA that has the anticodon of AUG will carry the specific amino acid that is methionine. So what will happen? That RNA has a codon. With this codon, the tRNA will bind with the anticodon loop as it is anticodon. So uh, interaction, a hydrogen bond will take place and it will bind to the RNA, mRNA, and it will carry the amino acid that is specific for that codon. So this is the function of tRNA. As tRNA transfers the amino acid to the RNA, so it is called a transfer RNA or tRNA. Now, first, how the amino acid tRNA synthesis takes place. That is how the amino acid binds with the tRNA. So, first thing, when there is no amino acid attached with the tRNA, we will call it a uncharged tRNA. When tRNA is attached with the amino acid, we will call it a charged tRNA. And an enzyme, that is amino acid tRNA synthesis, helps in binding of this amino acid to the tRNA. And where the amino acid binds? Amino acid binds to the acceptor arm. So through the enzyme amino acid tRNA, and amino acid whose anticodon is present in that anticodon loop will bind with the tRNA. So now first come what is peptide bond. Roger has already said yesterday that two amino acids attached with each other by a peptide bond. So say this is one amino acid, this is another amino acid. So whenever we will add two amino acids, then two amino acids will add with each other by a peptide bond. C double bond O NH. This is the peptide bond marked in light blue. So this is the peptide bond. So you can understand if you think for some time, taking some time, that one tRNA is carrying out one amino acid, another tRNA is carrying another amino acid. So how this amino acid will bind with each other? By a peptide bond. And how it takes place? So say this is the tRNA. Here it is written peptidyl tRNA. That means what is peptide? When more than one amino acids are attached with each other, we will call it a peptidyl tRNA. Okay. So more than one amino acid are attached with this tRNA, peptidyl tRNA. Now a new amino acid tRNA has come. Okay. So what will happen? This NH2, also a nitrogen has free electron. I have said in the first slide that free electron are the main culprit of every reaction. So this free electron will attack the carbon and this bond will break, and this tRNA, the previous tRNA with which the peptide was attached, will become an uncharged tRNA. That is, now with this one no amino acid or peptide is attached. It is an uncharged tRNA, and now the peptide bond has become associated with the newly coming amino acid tRNA. And so now this amino acid tRNA doesn't contain one amino acid, it contains more than one amino acids. So we will call it, now we will call it a peptidyl tRNA. As it contains more than one amino acid, we will call it a peptidyl tRNA. And again, a new amino acid tRNA will come, and the peptide bond will shift to that tRNA, and that amino acid tRNA will become the peptidyl tRNA. You can understand that. So, how this translation takes place? The very important thing that helps in translation is the ribosome. What is ribosome? We all have gone to the in the previous days also. What is ribosome? Ribosome has two subunits. One is a smaller subunit and one is a larger subunit. And it helps in the translation procedure. Without ribosome, the translation cannot take place. And in the ribosome, there are three sites known as A site, P site, and E site. Why it is called A site? Because in this site, amino acyl tRNA binds. As amino acyl tRNA binds, so it is called A site. In the P site, the peptidyl tRNA binds. So as the peptidyl tRNA binds, it is called the P site. And in the E site, it is called the X site, where the uncharged tRNA, in the previous slide, I have shown that whenever the peptide transfers from the previous tRNA to the next tRNA, it becomes uncharged. So through the exit site, the uncharged tRNA exits. So it is called the exit site. So now how the translation takes place. Roger sir, is the video visible?
Yes, it is, sir. The video is clearly visible.
Okay, thank you. So here it is the mRNA. And I have said in a mRNA, in the 3' end, there is poly A tail. That means numerous numbers of A are together to form the poly A tail. In between the poly A tail, there are defined codons, mRNA codons, that will code for the defined amino acid. And in the 5' end, there is methylated cap. So what happens? First, the small subunit comes and it scans through the mRNA. And whenever it finds the proper codon, that is the start codon. What is the start codon? AUG. And with the start codon, the tRNA comes and binds. So the tRNA that have the anticodon of AUG will bind, and it will carry the amino acid known as methionine. So methionine carrying tRNA will bind. So what will happen? After this, the large subunit will come and bind to form the whole ribosome. And now this is associated in the P site. Now what will happen? The A site is still free. In the A site, an amino acid tRNA will come. And as I have shown earlier, the previous amino acid will transfer to the next amino acid tRNA. And that this tRNA, which has become uncharged, will lose from the active site. And this will, the peptidyl tRNA that have more than one amino acid will move to the P site. The newly coming tRNA that has one amino acid will come to the A site, and like this way, the peptide formation will take place. So whenever new amino acid will come, amino acid tRNA come, the peptide will transform to the new amino acid tRNA. Until when? Until it comes to a stop codon, because stop codon doesn't code for any amino acid. So here a release factor will come, and whenever the release factor will come, the ribosome will dissociate, and we will get the peptide or the protein. So you can understand, from starting from the DNA, we have synthesized the protein, and this complete process, this complete way is known as central dogma. So with this, I want to stop my talk. I have talked a lot. So thank you.
Thank you, Dr. Somsubhra Thakur Choudhury. I'm simply spellbound. I cannot comment anything. I should not comment anything on this, marvelous lecture. It was like I am sitting in a cell, and whatever the going on with the DNA and RNA and protein, everything I can visualize in my eyes. So it is seriously a very, very, nice lecture and very good demonstration. So I
Thank you.
must congratulate Dr.
Thank you,
Thakur
sir.
Choudhury. It's a seriously excellent experience for me. So now you just take some rest, because
Yeah.
you have done a, very nice job. I would like to now call upon Dr. Mohit Mojumdar. Dr. Mojumdar, are you there?
Hi, Rajat. Can you hear me?
Yes, yes, Dr. Mojumdar, welcome, welcome. So, to my all the audience, I would like to introduce Dr. Mojumdar. Dr. Mohit Mojumdar is the Director of Business Development, Fine Biotech. He is actually associated, very closely associated with our department from a long back. And Pine Biotech is also a very close, in a association with us. Pine Biotech offers several courses on bioinformatics. Our students usually, avail those courses, and Mohit Majumder is a very close friend to mine also. And he is a very good speaker. I have listened a few of his lectures, so I hope that you all people will enjoy his lecture also as well. Professor Thakur Choudhury's lecture you enjoyed. So I'm pretty much sure that Mohit Mojumdar will give you a very good knowledge about the bioinformatics, what the bioinformatics is. As you all know that in today's world, without computer, we cannot do anything. From the morning to the night, whenever we are waking up from the bed, and till whenever we are going to the bed, everything is done by the computer. Somehow we are related to computer. No matter how much educated I am, no matter how rich or how poor I am, whatever it is, what the earning process I am doing, everything is somehow related to the computer. So definitely biology will not also be apart from that effect. So biology will also be somehow related to the computer science. And when these two subjects, two sciences, biological science and computer science merge together, that forms actually the bioinformatics. I think Dr. Mojumdar will speak more about that and he will explain more nicely. So without losing any time or without wasting any time, I am just requesting Dr. Mojumdar to start his lecture. Dr. Mojumdar, please.
Thank you, Roger. Thank you so much for this kind introduction. And thank you, Dr. Thakur, for this excellent talk. And I think it actually makes sense in the context that what I will be talking is very much related to his talk. So, yeah, as Roger said, that let's not waste more time and start my presentation. So let me just share my screen and please let me know if you can see my screen or not.
Yes, yes, Dr. Mojumdar, it's perfectly is visible.
Perfect. So hello everyone. Welcome to today's session on Introduction to Bioinformatics. So before going into the session, I just wanted to give you an introduction about biology. As already Roger said about the aspect of computational studies, aspect of data generation, how it has changed biology. So biology as a data science. So biology is rapidly acquiring the character of data science. Billions of data points on genes, proteins, and other molecules are compiled in large files and systematically studied. So, analyzing this data will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crop, livestock, like it affects almost everything. So, my name is Mohit Majumder. I'm one of the global representatives of Pine Biotech. Pine Biotech is a bioinformatics company that is working towards providing cutting edge research and education for students and researchers in India, US, Africa, and around the globe. So, our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform. We work with students and researchers, provide research support, big data analytics tools designed for biomedical discovery. So, ultimately, we are working for a future where we can contribute to enhance human health and well being by enabling biological research and discovery with data solution and support. So I'm really honored to have been working with people that have a diverse experience in this area, including our CEO and co-founder Ilya Brodsky and other co-founders of the company, Dr. Alfred Trauber, who is a philanthropist who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. So he is the core of, he started the Trauber Bioinformatics Research Center. It is one of his projects. So at another co-founder of us is Dr. Leonard Brodsky. He's the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics and over the course of his career has developed multiple novel algorithmic approaches for the analysis of biomedical data analysis. So these methods have been widely used. On the right, you could see Julia Panoff, Avi, me, Joshua, and Deepsha. We've been working to develop this platform into a educational, resources, provide educational resources for students, collaborating with scientists around the globe and helping them with their research. As I was telling you, we are a US-based company working closely with several universities across the US, collaborating with Thakur Bioinformatics Research Center, and developing these tools and programs and courses like Roger said. We already have a collaboration with SNU, and we did this data science for biologists course last year, which was very successful, and we hope to develop more collaboration and learn more about the students and the needs of the faculty. Coming back to today's topic, today we will try to understand the application of data-driven biology and application of bioinformatics in various areas of research. So I saw that you guys have just finished this lecture, which is like the core of. There is no running away from learning the basics, but there is a vast importance of what is happening today in terms of translational sciences. You already have learned that how the new science policy or the new educational policy has changed, and we are now more focused on developing solutions using science. It's just not doing the theoretical science, but also applying this science into discoveries, applying this science into products, applying this science into pharma. So there are several opportunities that I today will be talking about. So to start with, I just wanted to say that bioinformatics skills are required for anyone seeking to participate in advanced life science research projects nowadays. And if you see like how it goes, like you start from like when you start learning about science, it's a long journey. That's for sure. A research is something that you develop over the years. But I mean, what is the right time? Shouldn't it be good if you start like really soon, so that you reach that level where we require you to do translational stuff, where we want you to work on vaccines, where we want you to work on and like contribute to the society by working on like different problems, for example this pandemic itself. So a big challenge for those interested to get into bioinformatics or data sciences or the data driven biology is to get started. That is a big challenge because it's like it's not standardized here. So that is what we are trying to do here. So once you get started and getting involved and feel comfortable using basic tools, you can easily continue your journey navigating through complex and dynamic world of bioinformatics. So before talking about bioinformatics or just generally about sciences, how this has evolved into the field that we are seeing right now, because of what has already been happened. So if we recall, it has been 70 years since the structure of DNA was discovered. 50 years since the last time sequencing we did the sequencing. So it has been like a long period of time where we have been developing and learning about all this technology and all this information that we talked about. Dr. Thakur talked about amino acids and nucleotides. So how we learned about those? So it comes through a long journey, but today we have a lot of information. So in 2015, 1,000 genome project brought a significant number of whole genome sequences into the research community. And what is this whole genome? What is how this technology works? I will be talking about in today's class. So as you see, like in 1950s, we have had the DNA structure, and then after 50 years, we had the Human Genome Project. This was a very, it's a landmark project. We spent almost $3 billion US. If I say we, I mean humanity. So we were hoping that after the Human Genome Project, we will be able to address most of the problems associated with diseases, why this happening, why this viral outbreak, how to solve the issue with vaccine development, and also drug designing and development in general. But what we realize is that the science is way more complex than what we think it is, because we thought that knowing the sequences will do. But now we understand the sequences have a really complex role when you put together in terms of how they operate within the organism, or within a host, or within a specific cell. So how these technologies are helping us understand this complex nature of cell, and the complex nature of dynamics of the cell. So there are multiple ways we are generating this data. Okay, so omics data can be generated using NGS, and this technology explores the roles, relationships, and the actions of various types of molecules that makes up the cell of an organism. So NGS, or what is also known as high throughput sequencing, to start learning about this type of big data, the opportunity and the challenges they come with, we will be showing you the resources that are associated with them. But to understand the terminologies in itself, phenomics, genomics, epigenomics, transcriptomics. So what are these technologies? Phenomics is something that we observe. For example, if I give you a really clinical example, when you go to a doctor, I mean, when you visit a doctor, the doctor asks you about your age, right? And about maybe checks your weight. So those physiological factors affects the drug you're taking, right? That's the reason he's asking. He's seeing that whether you're a male or a female, and then also that classification is also something that helps in the diagnosis. So how this technology has evolved into something more deeper, and it has to be deeper because these diseases are way more complex than we thought. So we don't have a vaccine for HIV yet. The virus replication rate is really high. We don't have solutions for all the cancers yet. So what is the reason we have not been able to do it? The reason is that because we don't have the information, or the all the information that is required to know how things work as inside. We are gathering information from years now, decades, but the information that is required to understand the whole picture is something that we have started to do now. So the data, as I was talking to you about phenomics, phenomics is a study of those phenotypic characters, or phenotypes, as the word that you might have heard a lot. Genomics is about looking into the genes, the sequences, and how it affects. Epigenetics is a study where we are looking at DNA modifications. Transcriptomics is something when we are looking at gene expression. So for example, there is a state where a person is having there is a normal person, and there is a person who is having cancer, right? So what we do is that we collect a sample from a normal person as a control, and the cancer patient, from the cancer patient, we took some samples, and we see how the proteins are expressed at that point of time, like a person having cancer, a person not having cancer. So when I'm saying proteins or genes, so 20,000 genes in one sample, one person, and like another 20,000 genes in another person. So what we are trying to do there is we are trying to compare and see like which genes are expressed, like highly expressed, which genes are significantly uh, less expressive. So by that we will be able to understand that the targets, or maybe the genes that have been the proteins that have been changed their expression, and why. Those are the questions then we ask, and then we deep dive into the problem and identify that okay, this is the reason. So that is the kind of research that is happening right now. in around the globe, and that is helping us transforming science in and developing that understanding. So before, there's a hands-on aspect to every webinar that we do. So the hands-on aspect would be to, go to the site. This is an educational platform that we have created that I was talking to you about, that to help people understand, learn about these technologies, which are not so easy to learn because of a couple of challenges that I told you. One of the challenges that, you think bioinformatics to be something to do with coding. Many people, the courses starts from understanding of coding, so we eliminate that problem because we feel that the biologist who is working on the experiment should be able to understand his own data, should be able to understand. So you are the person who understands, central dogma. You know that you are doing an experiment, what kind of data is coming out of that experiment. So you should be the one who is analyzing that data as well. So to help achieve that, we design courses. So I just want you to go to this website now and register and take one free course, Introduction to Bioinformatics, which I'm asking you because it is just a very basic course for everyone. You does not need to have any knowledge to do this course. So maybe I can also paste a link here in the chat. Just 1 second. So that so if you can see my screen, you just have to create a profile in this site, and then you have to go and take this course. So this course talks about introduction to bioinformatics. So I'm just gonna tell you more about it, but just as for a hands-on exercise. uh, okay, there you go. So please visit this link and take this course. It might just take you 2 hours to finish this, so you can do it afterwards. So as I was telling you that the components of this course, so this is an introductory course that you can take. It's free, and it usually tells you about 2 to 3 things. The course includes introduction to big data bioinformatics, bioinformatics and healthcare, and translational bioinformatics. So you will be easily able to see the application of bioinformatics. Maybe also in agriculture, you will see the role of bioinformatics in defense. You will see the role how this bioinformatics is being applied in translational medicine. That is a very how pharma is using it, how what are the job opportunities that we have right now with these sort of skill sets. So it's not so distinct past where data generation was the bottleneck. So in the past, we were trying to understand by doing single experiments. So we were trying to understand a single enzyme. We were trying to understand about a single protein. We are trying to understand about the function of a single gene. So the structure to function or sequence to structure research is something that is what we are reading in our books right now. All the books that we have been reading about biology, about sciences, has been evolved through that kind of research and have come into the textbooks because of that. So in the current world, things have changed. But in the distant past, what I was saying is data generation was a bottleneck. And now the bottleneck has changed. The bottleneck is now data mining or extracting useful biological insights from large, complicated data sets. In the past decade, technological advancements in data generation has changed this entire thing. In particularly NGS technologies have allowed researchers to screen changes at varying biological scales. As I was telling you, like how a person is different from another person. So first of earlier we thought that that's like just 1% of the genome, the coding region is what makes all the changes or all the differences, and that's the main thing. I mean, exons, we would just want to focus on exons. Now, I mean, we understand that that 99% plays a huge role, that no non-coding RNAs and all that research is now bringing the importance of the other half on the more major part of the genome. So we used to call it junk DNA, but why we used to call it junk DNA, because and still call it junk DNA, because I think we don't understand the way it all works. We don't understand many things, or we are not able to correlate also many of our capabilities to the genome or to the molecular level. But how we are reaching there? We are reaching there by now collecting data. And how we are collecting data, as I was telling you about time point. So if you get sick in 3 months, you collect your sample and you collect your data and you see yourself, and then you get well, and then there's a series of events that's happening with one individual. So if we collect data on all those events, we will be much more aware of what is actually happening, maybe because we will be able to compare stuff. We will be able to see that this state one, this is state two, so things like that. But I mean, easier said than done. So because it's a big data, so we are talking about 3 billion base pairs. So genome-wide genetic variation. We are talking about those point mutations that are changing everything. Those point mutations are causing diseases. Those point mutations are, changing the characteristic, one particular trait. Gene expression, I already talked about gene expression. And then small RNA, I talked about small RNA, epigenetic modification, protein binding motifs. So all those things, like the chromosomal conformation in high throughput, is something is worth looking at, is worth understanding, because it will definitely gonna solve a lot of challenges and a lot of things in the future, and it's coming up really well. So if you go into this course, you will see that there are videos that explains some of the things that I've talked about today, and there is a quiz. So once you're done with it, you have to finish the quiz. Introduction to bioinformatics can be completed like in very small time. Quizzes help reinforce the learning material, summarizes the topics that we covered. So you can also use like glossary of terminologies, like you can click here, then it will show you what it is. So it's pretty easy, and something to check out. Now, I was talking to you about data-driven discovery. So data-driven discovery involves several steps. So in terms of, thinking, like just thinking about research. So when a scientist does research, when your professor does research, when all around the globe it's happening in the hospitals, where do we start from? Have you ever thought, like, where do we start from? Does that happen? So it happens through knowledge, because first we have to learn about one thing, that what is out there, before trying to do something new. So if you are trying to do something new, you have to know what has already have happened, because or else you end up doing the same thing that has already have happened. So literature review is very important in that aspect, and knowing what is done and what can be improved is something that you can start thinking about. That's how the research aspect of things work. Like, we know that, okay, this is a problem. Now how do we find solution to that problem? So identifying the question is very important. Review finding, design model, gather data. So what we do is that we look into a problem, and we see that for example, COVID-19 has generated in 1 year so much data that no disease has ever. Like, it's everywhere. Every country, there are bigger labs working on it. They are generating data, And now we have so much data on COVID-19. Now we think that we still need a lot of data because coronavirus was not well studied before. So we started doing it now. But yet, I mean, we don't have a solution yet. Why you think, I mean, with all this people working and all this information, why you think that it's has haven't been, happened yet? And why we are taking this time? So this has been the challenge in, biomedical industry, challenge of doing drug design. Usually it takes 15 years, 20 years to design a drug. And that was because we were using a lot of high throughput, not sequencing, but screening. So we were trying, hit and trial kind of a thing, that whether it works or it doesn't work. And then there are side effects. So when you're talking about a targeted study like this, then it makes more sense, because individually we are all different. So we should have a personalized, personalized way of handling and dealing things. So as I was saying, that every one of us are different in the sense that we perceive things differently. Our body at a molecular level is different, and also the environment that we go through, like the micro environment, the metabolome, does also play a very significant role. So how do you understand all these things and think in the context of one person is a thing to be seen in the future, and it's coming up. So really exciting times in that context. So from data to discovery, researchers, are generating large volumes of biological data, which has changed the way the biomedical research is done. So the volume and heterogeneity of the data are limitations right now that that is challenging our. That is the challenge, and machine learning is playing a role in here because of this. So why machine learning suddenly become so much such a big thing? Why data science? Why people are talking about data science? Because you see, like everything is online, and we are generating like megabytes of data maybe daily. Like if you are scrolling your phone, if you're scrolling your website, you're posting your pictures, you're buying on Amazon, everything is getting recorded, and every bit of that is data, and it's getting bigger. So challenges for the company is to analyze that data, and they're doing well. They are using that data, using machine learning techniques to understand their users. And similarly in biology, what we are trying to do is that we are generating data, at least in the developing countries or developed countries, they are doing it quite efficiently. So there are different steps of designing an experiment, but that you can learn more in this course. So I will not waste more time into this, but to see that this is like like a big cycle. So it's just not like just generating data and relying on it. So there are several elements, just not that. Biological interpretation. So once you understand that, okay, the importance of learning what you're doing right now, as in microbiology and biotechnology, is going to be helpful for your entire course of your career. So whatever you're learning right now is very important. Those books that you're going through is going to help you develop that analytical understanding of how things work. So once you are focused on a single problem, then you of course can just focus on that one problem and do more and more about that one problem. Then you're very you're working on a niche problem, then you can do it because you're not reading about everything around it. So that's the way to go about research, specifically to target something very niche, and then work on it, because there's so much out there. It's really gets overwhelming at the point of time, and we lose interest. So the application of this kind of sciences. So as I was telling you that how this is taken out like from samples, but there are databases that are publicly available, freely available for you to go and look into all of that, like the TCGA portal. The TCGA portal has like a lot of information on all kind of cancers. There, genomic data, you will see genomic data, you will see transcriptomic data. You will see that you can compare a normal person or samples of breast cancer, samples of liver cancer, ovarian cancer there, and you can start working on it right away. It's like that. So that has changed. That has changed, which was not happening before. What was not happening before? There was only limited number of people who had this kind of an access and the tools who can do the research. Now, with the repositories being publicly available, and companies like us who are helping people to understand and learn from this kind of research, it becomes really easier for even students to do research and publish. So the translational aspect of this is to be able to find a personalized treatment, as I was telling you, or what is a personalized treatment? So here in this example, you can see what is happening here is that a sample is taken from a patient. So this is a cancer patient. So this tissue is implanted in a mice. So atomic. So there are different kind of mice models that have no immune response. They're designed for the scientific experiments. So I think it will take me like maybe another lecture just to talk about the mouse models. But let's not do that today. But just tell you that to understand the effect of drugs, and we experiment on like mouse models, and that is the tissue is being grafted and in this mice, and then we are testing on different cell lines, that how it is on different cell lines when we put this cancer cell and seeing the effect of a particular drug. So we're trying to do is that based on this information, we are doing molecular profiling. So what is molecular profiling? We are separating people based on their molecular profile. So we actually already know, for example, in breast cancer, there is a panel called PAM50. So in that, what is happening is that we know in breast cancer that these 50 genes, or there are some genes in that 50 panel of genes that are overexpressed. So we know their expression from that these are the genes that will be overexpressed in breast cancer, in a specific type of breast cancers. So those genes or proteins are called biomarkers because we already know them. They are the markers, right? So that is some of the application of this kind of uh, mm, uh, technology. So talking about the platform, once you learn all this, you can get a certificate by finishing, and you will have an academic profile on the platform, which means that we have almost 5,000 people around the globe, many from top universities in the US and top universities in India and many places. You'll find people in there. You can talk to them, collaborate if you find them interesting, and you will have a profile of your own when you finish this course and enroll for this course. And you can put your pictures and stuff, and your social media links for communication. So this is the certificate that I was talking to you about. Okay, so if anyone have any questions on whatever we covered, please feel free to ask. And now I will just build on that, what we have discussed, and show you the rest of the presentation. So no questions. Very good. Okay. So
Dr. Majumdar, maybe the questions will come later on in the chat box or by email. So we will definitely
sure,
communicate with
sure,
you.
sure,
Yes.
sure, not a
Hmm.
problem. No, I just wanted if there is any question now. So that's okay. I'll be happy to answer those questions. So many of us here, as I was telling you, many of us are here are biologists that were trained in basic biological processes and then specialized in a certain area where we gained both knowledge and experience from experimental data. Many of the examples we learned from information we accumulated came from carefully designing experiments where there certain assumptions were tested. We were testing an hypothesis. The generated data from those experiments confirmed some of the assumptions, and those were recorded as proven hypotheses. For example, from a biologist perspective, we understand cancer as a set of changes on a cellular and a subcellular level, causing a specific clinical outcome. So once we design an experiment to check for an expected change, we can start associating the molecular change with the phenotype, a clinical manifestation. Ideally, we hope to map out those relationships that we found and make sense of them. So this is a typical way of data connection between the data. And when you trace it back to the pathways, molecular pathways, and you understand identify the key players, identify the pattern, and learn how to manipulate them to find a cure or cure for a condition. That is the challenge, and that is what we are doing in this. So in reality, however, many biological phenomena do not play out according to this formed rules and formulas, because that's what I was telling you, that it's way more complex because of the involvement of multiple factors. But now, after the Human Genome Project, before that we were doing the single experiments. Now, after the Human Genome Project, at least now we know better. So the big data. So when I was just talking about the big data, and it's really difficult to visually or to understand how big it is, because when you see a really tall building, it's big because you can see it for sure. But when we are talking about data stored in drives, we don't even realize like what it is. We just maybe underestimate it. But it has a few qualities, five qualities that define the big data: volume, velocity, veracity, variety, value. So which is ever growing. So a biologist is largely, driven by a hypothesis, which is often a result of past observations. These observations give rise to a question that can be identified. So it's just like a summarization. I'm summarizing the aspects that we discussed today. So the observation give rise to a question that can be definitively answered using data generation generated in an experiment. This hypothesis can be confirmed or refuted after you analyze the data, and you have come to a conclusion whether you want to support the hypothesis or refute the hypothesis. So the collection of results are knowledge. But big data is changing this way we view results, as I was telling you, that because what is happening here is that for example, this part is already done. For example, like people have generated data, they have deposited it in NCBI or in TCGA, all those repositories. Like NCBI has humongous amount of data, like insane amount of data, but not that all the data has been analyzed, because some people have generated data for a different aspect, maybe for a different biological question to answer, but that data can be used for answering another question as well. So NCBI, as I was telling you, has grown exponentially in the past few years. Just to tell, the recent growth in high throughput sequencing of cancer genomics and transcriptomics has created a lot of big data problems. So some of the resources and repositories are like this: Array Express, TCGA, as I was talking to you about. So where you can get sequencing like RNA-seq data. So big data and precision in oncology. And I was telling you that identifying a patient and patient stratification is something that we discussed. I know these are like big words, but these are simple to understand. If you go through maybe a learning experience, take the Introduction to Bioinformatics, you'll understand that this is something that you may be able to comprehend. So the big data problem preludes many cancer biologists and oncologists from gleaning knowledge from data generated regarding the nature of this malignant processes, as well as the relationship between tumor genomic profiles and treatment responses. So how a person is responding to a treatment. I mean, if a drug is given, then if we are looking at a genomic level, that how it is helping in targeted cancer therapy. So talking about this therapies and how this entire paradigm has changed, how the pharma is using this, how this big pharmaceutical industry, the small biotech companies are working. So genomics is now considered as a legitimate big data field. Just one whole genome, human genome, produces approximately 200 GBs of raw data. So one genome is approximately 200 gigabytes of raw data. So if we manage to sequence maybe 100 million people, which is happening in many countries, so 100 million people by 2025, we will have what? We will have 20 billion gigabytes of raw data. So that's massive, right? So when you have this sort of data sets and databases, you need maybe a computational infrastructure to be able to, or even to understand how they are connected. So just to say, like, acquiring bioinformatics skills is in today's world is of tremendous value, because if you see the postdoc positions that have been currently offered, or how industry is being evolved, I think a lot of job opportunities are there. But you probably know that the online courses certificates are not yet valued that much. I mean, one of the reasons for this is due to the lack of maybe community oversight and mentorship. How many times? I mean, I have I have this experience. So how many times? I mean, I have gone through Coursera edX and felt sleeping watching a video. So I think one of the things that is with this online way of learning is to be able to interact like the way we are doing it right now. So once we are doing it through like fairly regularly, I'm very sure that you'll ask more relevant questions and understand this into more depth. So I think this hybrid way of learning is really nice, and that's why we have Hello, can you hear me? Hello.
Yes, Dr. Mojumdar, now we can hear, but last few slides we cannot hear actually. That's why I
Okay.
was trying contact with you
Yeah,
from the.
someone muted me here. Some Ravi muted. So okay. So can you tell me from where
Yeah,
I
yeah,
stopped?
that thing
This one?
about the introduction to genomics. No, no, no. After that, no, after that, from the
Metagenomics?
yeah, introduction to genomics. From this slide, actually, you have got muted.
Oh,
Yeah,
okay.
yeah.
Sorry about
So
that.
please resume this a few slides once more.
Sure, sure. So
And
as I was saying before, that these courses are free. So Introduction to Genomics is a free course, and as you have learned in the previous class, every aspect of central dogma, and then what is DNA, what is ATGC, and how these mutations are causing are happening, and the variation, like why we are different. I mean, why how a mutation plays a role in that. So Introduction to Genomics is a course which will help you visualize genome chromosomes on NCBI browser. So it will be, you will be asked to open another browser, and there will be a file which you will click, and it will show in the browser that how you can see this entire genome, the chromosomes, and the segments of genes in that. So please, it's a free course. I'm recommending you to take it. Introduction to Metagenomics again is another free course, which covers metagenomics. So microbiome and metagenomics, how they are playing a role. So I talked about environment, how environment plays a significant role in how we are reacting to different this maybe in terms of medicine, how drugs are affecting us. So to understand that, I mean, people are studying the role of gut microbiota. So how the samples taken from our gut and the mixture of bacteria and all the different microorganisms are there can help understand more about our body and how it influences our body. So human gut American gut project is a project that happened where they've taken samples from a large community of people and did that analysis. So it gives you an overview. So this is again a free course. Introduction to Epigenetics, to learn about epigenetic regulations and changes and DNA methylation. So different type of all these changes that are happening within the cell, within the DNA, which is not a permanent change. So to learn about epigenetics, transcriptomics one is not a free course; it's a hands-on course. So when you do transcriptomics, I mean, after you are like well versed, and if you find this thing interesting, so in transcriptomics one, what you will learn is that the gene expression. We're talking about cancer patients. We talked about cancer and normal cell. We talked about the PDX, the mouse project. So in transcriptomics one, what we do is that we learn about what is transcription translation, why we learn RNA, why we want to learn about RNA, and how we can quantify RNA. Like from before, we used to use northern blotting and microarrays to understand and quantify RNA. Now we are using NGS technology and getting the gene expression. So I gave you an example. So that example is being covered in this, in the context that it is application of what you have learned, plus you have assignments, you have hands-on tutorials to work on the T-Bioinfo server. So the T-Bioinfo server is a cutting-edge platform designed and developed for students and biologists and clinicians. And why I'm saying this is that because maybe I can give you a demonstration. So is it visible, Rajat? Is this page visible?
yes, yes, Dr. Mojumdar, it's visible.
Thank you. So this is the T-Bioinfo platform, and this is an intuitive platform, which means like when you if you want to learn that how things, it looks really complex, and it is complex, but we want to help people understand this complexity easily. So to do that, for example, I will give you a small example today about. Maybe I have to log in first. So this login is not for everyone, but for people who are a participant, or like if you are working with us, something like that on a program or something. So however, this platform is free to use, like the Introduction to Bioinformatics. So this is, for example, this, you can say that this is our books. So this is my book, and this is the workbench. So this is where I do the experiment. So to learn about these experiments, I'll just show you a demonstration pipeline. So this is the cell line project, and you can see this is a complex bioinformatics, workflow. It has a lot of algorithms that do different things, and they are placed in a logical manner, so that when you input the data, it goes through this process. So it is first pre-processing, and then it is cleaning. The cleaning stuff is going on. Then we are aligning it to genome or transcriptome, and we are looking at the isoforms. So I did not discuss about the isoform problem today, but that would be another session. So what we are doing is, here we are taking samples from a cancer patient and normal patient, and we are putting it inside this inside this workflow, and see how we can get the table of expression. So what is happening, what happens in NGS, when you do this experiment, NGS experiment, you get a raw reads. You don't get like table of expression. So from raw reads to table of expression is a complex experiment. So we can do it easily here. And why I'm saying easily, and it's meant for everyone, because when you click start, it shows like what is there, and it's a point and click system, so you don't have to code and do all those things to get this kind of experiment running. So on also, this is an AI guided platform. So what is happening here is that when I click start, it has highlighted me a path that based on this data I should use Trimomatic. So Trimomatic is something, is an algorithm that helps us to remove the technical sequences. So what happens is that in sequencing, in NGS sequencing, when we are putting that sequences into the sequencer, we are we are uh, joining adapters to the sequences. So those adapters are like sequences that comes and goes and ligate at that 5' end, and those are the technical sequences. Those are not our sequences that is coming from the samples, but those are for the experiment to happen successfully. So to do that afterwards, like after when you get the raw read, it will come up with all that noise that have been that is the limitation of the experiment. So we are not getting a pure result of sequence. So we use a lot of statistics and mathematics and algorithms from that state of raw data to a biologically meaningful data. So that is the whole process that you will learn in Transcriptomics 1, that how we will be able to solve and understand transcriptomics data analysis. So that is one of the aspects of transcriptomics data analysis. And this is the entire process. So once you are generating a sample from a sequence, or COVID patient in Kolkata, for example, you have the genome. Now what to do with that genome? You have to clean it up. It's coming from the machine, it's a raw, and then you use platforms like this, or you develop a platform where you can do this sort of analysis. And then once you have the entire genome sequence as a FASTA, then you can do further analysis. So in that context, this is a point and click system. As I was telling you, that I will click on Trimomatic, it will tell me next I have to click here, and I will be able to know everything about this algorithm. It's described here, and there is also publications that are also associated, that are also put in here. So I will do So if you can see, there are publications. If the algorithm is published, you can find the publication and learn about the algorithm. Input output files is a demo pipeline. So this is point and click, means that you have to just click and you can go back and change that path and stuff. So once you do that, you have the file as an output. So this is for teaching. We have built this platform and this this demo pipeline for faculties to teach, and also for us to teach. So it's, what we did in here is a big experiment, but we will be able to understand through like different sessions and understand that what is this isoform expression, what is this gene expression, and how we analyze this and understand whether they will be biomarkers for cancer, whether there will be a drug target for cancer. So these are important, very important questions in terms of biological research, or individual research, whether it is in agriculture, whether it is in defense, or whether it is in different aspects. In understanding the mechanism of an enzyme, also, for example, not an enzyme, but mechanism of a disease, mechanism of a behavior. So there are multiple factors involved. So we use machine learning for that. So that I think I'll not be able to cover in this one, but you can obviously learn all that using the platform. So as I was telling you, that this platform is developed by Tauber Bioinformatics Research Center, Haifa, Israel, and we have several collaborators in UCSF, Cambridge, that helped us, with several of these pipelines to be designed and being able to be used by the scientific community and students. So the state-of-art design helps us to go through this and do a high-quality research in a smaller amount of time. So that's one of the benefits. And machine learning is something that we use for the big data, because it's very difficult to understand the pattern. If you, for example, you can plot, two genes. I mean, you can plot, like, for example, mm, uh, age. It would be better to give you a visual, so maybe I will skip that. So just to give you an example, like it's easier to plot data into XY and XYZ. So two-dimensional, you can plot data and understand the analysis. You can plot three-dimensional graphs and understand the trend. But when you have thousands, when I'm talking about thousands of genes, then it becomes a problem of dimensionality. So a problem of dimensionality is that it's not human readable. You cannot plot 4D graph or 5D, 6D, 7D graph, but machine learning can. So why machine learning is helpful is because from this huge data, machine learning is able to give us the important data. So understand the variation. When we are comparing, for example, a normal person with a cancer patient, we are comparing. Always remember that we are comparing, and when we are comparing, we have to compare the entire both of them together, and that's a computationally a challenging thing to do when you have not hundreds, not but thousands, and maybe more than that of number of samples. 100,000 genes you have to look, and you have to find maybe 20 genes out of it. How would you do that? You would take help from machine learning. So as I was telling you, we just did the course previous year, and we are doing different online programs to help students and our universities, collaborative universities and research centers. So these online programs, the Omics Logic programs, have three components. We do Zoom meetings and this asynchronous courses that I showed you. So when you take these courses and you get also points for doing that. So you will see that how much you have progressed and how much you have learned, and also you get certificates that could be that gets stored in the profile, and that could be shared, and it's always there. So you can put it in your LinkedIn, or you can show it to your recruiter or whatever. So another aspect is the practical assignment. So we have come up with a Python and R tutorials and courses. And it's in a similar way, we have designed a platform so that people can easily learn Python and R and implement those kind of skill sets as well. So I would talk about a couple of programs that we do. Omics Logic Transcriptomics is a 1 month program, which we are going to start on 16th September, where we will talk about entire transcriptomics, data analysis. So transcriptomics 1, transcriptomics 2, 3, 4. We have 4 courses. One talks about the entire, raw data to gene to the table of expression. And when I say table of expression, it means the genes, the list of genes that have the expression levels in numbers. So you can just read in numbers, see in numbers that how a gene is more expressed and how a gene is less expressed. So in transcriptomics 2, what we do is statistical analysis. Transcriptomics 3 is machine learning. Transcriptomics 4 is single cell transcriptomics. So that is again another advanced, aspect of learning how to interpret single cell data. Metagenomics, again, about the microbiome and about understanding how these viruses, how these small organisms are affecting the microbiome and affecting different cellular processes. Genomics is to understand and learn about genomic data analysis. Epigenetic, I have already told you about. So other than that, like the omics logic programs, we have like 4 omics logic programs you saw on data types. So those are data types. Now these are research programs, which means that we are addressing a specific research problems. Like for example, infectious diseases. We did a program where we learned about when we did a coronavirus, we took all the sequences of coronavirus sequence, and we tried to address many biological questions. And this was a really successful program that we are doing with students from all around the globe. People came up with bioinformatics projects from coronaviruses, different type of SARS-CoV-2 strains. People from Brazil, they came up with their own. Sri Lanka. So like it has been an amazing experience. People working from different outbreaks. So we are here analyzing data from different outbreaks and understanding the biological significance and what can be done. Precision oncology is a specialization track for precision oncology. Precision, which means that cancer. We're talking about different cancer. If you're interested in that, you can check this out. And bioinformatics for precision medicine is about precision medicine, which means that addressing the challenges and future of medicine, how this knowledge can be transformed into drug designing and understanding how we can have personalized, techniques to help individuals. We did a course on SARS-CoV-2, specifically on just for SARS-CoV-2. So anyone who's just interested in learning all the aspects and understanding how the spike proteins and how the sequence, from the sequence to structure to function. And this is a course again for data science. This is for statistics, Python, R. So these are specialization course, or you can say advanced courses that helps you develop skill sets that are matching to the industry right now. So the CodePlay, this third platform that we are launching this month, is for Python and R. So you can just put the code. Every help is there. You can run it from within that and generate visualization stuff. So when we have these projects, when we have the students finishing these projects with the university, for example, the students that finished the courses in SNU, we offered them, fellowship or research internship, where they learn about, or where they progress on a specific problem. So you learned about the data types, you learned about the examples where it has been implemented, you learn about the research, you learn about how it can help in translational kind of science.
Hello, Dr. Mojumdar. Cannot hear you, Dr. Mojumdar, again. Hello.
I think again the same thing has happened. Uh, Probably his net is not good. Connection loss is showing here.
Okay, so just let me call him once.
Yeah, it is because it is not about mute. Here it is clearly showing the connection lost.
Okay, okay, okay. So just let me call him once. Yes, I just talked to Dr. Mojumdar. Maybe he has some net problems, so he is rejoining again. Please be with us.
Hello.
Yes, yes, Dr. Mojumdar. Something went wrong again.
Sorry, when you lost me, what was the time?
What?
I mean, how long was I gone?
From this bioinformatics, data science, biological data, this slide, what is on the screen, from there, just you have got disconnected.
So my screen is visible, or I have to share again,
Uh,
right?
you are just showing the bioinformatics, data science, biomedical data.
Okay. So I was here.
Yes, yes,
Okay. So as I was telling you that after we finish the after one finish the program, like faculties or students, and they are, whether they are interested in research, they are interested in developing curriculums, to students interested in developing research projects. So once you have learned about the data types, once you have learned about the examples, once you have gained enough points on the server, then we invite people to work on research projects, and that is a bioinformatics fellowship program that we do. So other than that, that is another program, that is a fellowship program, that is for people who are like maybe in an advanced stage, like in third, fourth year, or maybe like doing a research project, or after learning everything, they want to work on their independent research project or have their own idea. In that sense, that research fellowship is helpful. So these are the some of the areas that we are working on: infectious diseases, data science, precision medicine. So this is programming and coding, precision oncology, so cancer, neuroscience, and metagenomics. So these are the research areas we have developed specialization tracks for students to learn. So research internship helps you develop, understand some of the concepts. For example, going through the omics data, learning about the omics data, mastering the T-Bioinfo platform, which I showed you, mutation variants, gene expression, metagenomics, biostats, machine learning. So this is all in, in a sequence, because first you want to learn about the basics that you're learning in your college, by your beloved faculties and very talented faculties. So once you learn from that, I mean, it would be nice if you can, understand some of the modern biology and the applicational biology and translational aspects. So these are some of those things that will help you understand. So as I was telling you, the people who finish, they do go on and publish, present their posters in conferences, and talking about what they have learned and how they have used this for their own research.
And another aspect of is to publish. So poster publications we have had from us. So this is why I'm showing you this, is because this is an outcome that is coming in maybe 3 to 6 months. That is literally very difficult to achieve for any, I mean, any institution. But with the help of these platforms and this very niche kind of an understanding, we were able to generate and people were able to finish bioinformatics projects and publish posters also in like F1000 research, research gate. So these are some of the benefits. And once you have well versed and done, you like these are some of the candidates. Hannah is from high school, so we have same programs from high schools to PhD and to faculty and to researchers. The same platform can be utilized by all of people needing to or have that enthusiasm to learn. So the most important thing is to be able to have that research interest. And being a biologist and microbiologist in SNU, I think it would be a really nice thing for you to start looking up and look up for these keywords, learn from your professors. You can reach out to us. So you have the opportunity that is a rare opportunity for anyone else. So please utilize that. And after you're done with all of it, and if you're confident, we offer jobs. So we have already hired two, three people, three students. So the jobs are, to develop curriculum or to train in omics logic programs where we take sessions like the omics logic programs I've showed you, and also to help students. Like all these programs have a component of mentorship. So the interns that we have are helping the students of our different programs. And now with the help of this collaboration between mentors, students, faculty, this entire process is very highly effective. So I will just end with this slide, and I'm open to questions. So please go ahead if you have any questions.
Yes, thank you very much, Dr. Mojumdar. Um, do we have any questions? Just let me check if we have any questions here or not. Maybe not in chat box or through email. We can have some questions.
Sure, sure. Please.
Anyways, whenever some question will come, we will communicate to you to address those questions. And you have explained a lot about the programs offered by your company and how the students can take the bioinformatics as their career. So it's a nice talk, Dr. Majumdar. I'm really very glad, and it's a very big opportunity for us to have you, a director of a company. And definitely, my students are now very much. I mean, they are a little bit of worry before that where we can go, where we can have our jobs. So they are now relaxed that we have fine marks for job opportunities, and Dr. Mojumdar is there. Okay.
Thank
So
you, and I hope that this is helpful. And thank you, Rogers, for inviting me, and thank all the dignitaries.
yes, yes. Actually, in online platform, we have to face some problems. Sometimes the net problem is there. Yesterday also in my session, the net got disconnected, and I was talking on my own way. So I beg pardon from all my participants that we are helpless. We cannot do anything. So please, forgive us for these kind of difficulties, technical problems, and be with us for next 2 days again. We have more sessions, good talks we are going to have again. So, is there nothing to more discuss? May I conclude the session with the special thanks to our two very eminent speakers, Dr. Mohit Mojumdar from Biotech and Dr. Somsubhra Thakur Choudhury from Sister Nivedita University. And I will thank you, Fatima Calcutawala, Professor Atrik Ghosh, and Professor Milan Boni for staying with us and helping this event throughout this 5-day program. So, okay. So I will conclude this session for today. And all the participants, hope to see you tomorrow again on the same platform at the same time. And the link again will be sent to you by tomorrow. Have a nice day. Stay safe, stay healthy, and bye for now. Goodbye, everyone.
Thank you. Bye bye.
Yes.